Okay, it is audible. Okay, it is audible. Okay, it is. All students, okay, it is. Likita Amshidhar Reddy. Message me, okay, it is audible. Okay. Okay, with regard to this, the biotechnology and obtaining gene product, obtaining gene product. As we introduce the recombinant vectors in the host cell, whether it is a plant, bacteria, or animal cells, and after having cloned these recombinant vectors into the cells, they are cultured on the medium and they are plated on what it is antibiotics. So, as it is having selectable markers, that is ampicillin resistant gene, that is said to be what it is selectable marker. If you link the gene at what it is tetracycline resistant gene. So, as you inserted the gene of interest into the vector and transferred to what it is the host cells. And ultimate aim is to produce what it is the desirable, the gene product, that is nothing but what it is the protein. You see this heading, obtaining the gene product. This is foreign DNA. This foreign DNA introduced into what vector? the vector, it became what it is, rDNA, recombinant DNA. And it is introduced into the host cell, that is bacteria, plant, animal cells. Why? For expression of what it is, the gene of interest. So, as we inserted the foreign DNA into the vector and transferred to the host cell for what? Expression of gene. So, the gene which got multiplied in case of what it is, host cell. So, gene multiplied in host cell. The desired gene multiplied in host cell. So, after having inserted this vector and introduced it into the cells, and we have to optimize the condition. So, ultimate aim in all recombinant DNA technology is to produce what it is, the gene product, that is the protein. So, ultimate aim in all rDNA technology obtaining desirable protein, that is gene product. Obtaining the desirable protein, the gene product. So, having cloned the gene of interest, introduced into the cells, bacteria, plant or animal, and optimize the condition for expression of the gene. For expression of the gene in case of the host cell, it required some technical steps. So we fulfilled all the things. Now I had to think of it producing in large quantities. Why had to produce in large quantities? It is available for what it is, so many number of what it is the people, to meet the demand of what it is the growing population. So I have to produce everything in case of what it is large scale. So ultimate aim in all recombinant DNA technology is obtaining what is desirable protein. So after having inserted the foreign DNA into vector, transferred the host cell like bacteria, plant and what it is, animal cells. So for what it is expression of the gene got what it is multiplied in case of what it is the host cells. And the production of the recombinant protein in heterologous host, whatever the gene for the production of the particular protein is transferred toward the host cell. So we are getting the recombinant protein in case of what it is other host cell, heterologous host, as gene encoding for protein in heterologous host. What do you call this protein? It is recombinant protein. So gene encoding protein in heterologous host called what do you call it is recombinant protein. So having cloned the gene of interest transferred to host cell, then we have to optimize the condition for expression of the gene for the product of what it is the gene product. So we have to think of it producing in case of what it is large scale. So after optimizing the condition or gene product, optimizing the condition. So which are some technical steps that is favorable condition for what it is gene expression. So there must be congenial condition for expression of the gene. We are providing the condition. So in that condition, gene is going to be what it is expressed. So after optimizing the condition for the gene product, the technical steps which are provided for favorable conditions which are provided, then you have to think of producing in large scale. Okay. So anything the biotechnological product has to be produced in case of what it is large scale. In laboratory cultures, only it is the small scale. In bioreactors, it is nothing but what it is large scales. So gene encoding protein in heterologous host called recombinant protein. After optimizing the condition for the gene product, so technical steps and favorable condition for gene expression, we have to think of it producing in case of what it is large scale production to meet what it is the demand of what it is our growing population. Suppose the cells having what it is RDNA. The cells harboring the cloned gene of interest cultured on a medium in case of the laboratory. In case of laboratory, it is the small scale. So, the cells harboring the cloned gene of interest, which are grown and which are the product it is extracted, purified by the particular technique. Okay. So, in case of the laboratory, the cultures are grown and product extracted and purified. In case of laboratory, what do you call this method? It is flask method. In small containers, what do you call it? The flask method. And there is continuous culture system. In continuous culture system, that means used medium is drained out from one point and fresh medium is introduced. As used medium drained out, fresh medium it is introduced, the cells kept in case of what it is physiologically active, exponential state to give large amount of what it is biotechnological product. Are you getting it? So, in continuous culture system, used medium drained out, fresh medium introduced. So, cells are physiologically active in exponential state to give what it is large biomass. So, in this condition, cells physiologically active, exponential state That means very fast growth. Exponential state, large mass of biomass. Exponential state, large biomass. So in continuous culture system, advantage is fresh medium is introduced, drained, that is used medium is drained out from the one outlet. So in order to keep the cells physiologically it is active, exponential state to give what it is large content, that is the biomass. What we call it is a continuous culture system. So using bioreactors is example for what it is continuous culture system. And if you take small volume cultures, it is low yield. Large volume culture, that is 100 to 1000 liters which are processed. 100 to 1000 liters which are processed in case of large volume cultures. For these large volume cultures, stainless steel vessels. For large volume cultures, it is the stainless steel vessels. The stainless steel vessels, what do you call it is? Bioreactors. For large volume cultures, the stainless steel vessels we are using, what do you call it is? Bioreactors. So bioreactors are stainless steel vessels where raw materials are biologically converted into products by using microbes, the plant cells or otherwise animal cells into product. Maybe food product, maybe veterinary product, maybe what it is the medical products. So in bioreactors, raw materials are biologically converted into products by using microbe, plant, animal cells in large quantities. So in these bioreactors, what are created optimal condition in order to produce the product. Temperature, pH, the salts, the sugars, whatever they required in order to give the particular product for the growth of the cells, we have to provide in case of what it is the bioreactors. Optimal condition, that is temperature, the pH, the oxygen, salts, nutrients which are provided in case of the bioreactor. So raw materials are biologically converted into products. As large volume cultures, that is 100 to 1000 liters which are processed in case of bioreactor. So continuous culture system, for this bioreactor it is the example for continuous culture system. Because user medium drained out, fresh medium it is introduced. So naturally cells are in exponential state, active state, physiologically active. So naturally going to give what it is the large product. So optimal conditions are provided in bioreactor. So in bioreactors, That is the stir tank. Bioreactors and sparged stir tank bioreactors. The two bioreactors, stir tank bioreactor, sparge stir tank bioreactor. In the first reactor, it is stirred tank bioreactor. In case of stirred tank bioreactor, and it is a stirrer, it is having blades. It is one type of the blade. It is another type of the blade. Okay. So in this it is pH control, that is acid base. It is steam for sterilization. And this blade, upper blade, it is flat blade impeller. And it is the culture broth. It is the culture broth. And through this, it is sterile air, it is passed. This foam, it is created. It is agitator system having stirrer. So in this stirrer tank bioreactor, labeling also is very, very important. This is the labeling. pH controlling system, that is acid base. Steam for sterilization. The steam for what it is, sterilization of what it is the medium, whatever the culture present in this. And sterile air, oxygen availability, sterile air is passed through from the base of what it is container. And in case of stirred tank bioreactor, it is cylindrical, curved at the base. As it is cylindrical, curved at the base, help in mixing up of the contents in the container. That is the one key point. And this stirrer facilitate even mixing and oxygen availability throughout what it is container. We are providing what it is the sterile air through what it is one, it is hole, that is port, what do you call the place? And stirrer facilitate even mixing oxygen availability throughout what it is container. Suppose if this culture broth it is stagnant, there is no availability of what it is oxygen. Unless it is agitated by an agitator. The agitator system it is having a stirrer. The stirrer facilitate even mixing oxygen availability throughout the container. Okay. And cylindrical curved at the base helps in easy mixing up of what it is content. Mixing up of content. Even mixing oxygen availability which is provided by it is stirrer facilitate even mixing oxygen availability throughout what it is container. And this bioreactor having an agitator system, foam breaking system. During the culture broth, the foam is created. In case of the vessel, the foam it break by means of what it is upper blade. So upper blade contribution is breaking of what it is the foam. So foam breaking system. And there is oxygen delivery system. And this flat blade impeller, it helps in what it is, that is circulation of culture broth. The circulation of what it is culture broth. Labeling is also important. Upper blades at the top of what it is the bioreactor helps to break what it is the foam. The blades below what it is upper blade, that is the flat blade impeller, helps in what it is, that is circulating of what it is the culture broth. Okay, and it is a sterile air. And over which it is temperature controlling system. There is jacket around what it is the container, that is temperature controlling system. There is a continuous falling of what it is the water over the jacket. So temperature controlling system, oxygen delivery system. And there are ports. The ports are nothing but the places, inlets and what it is outlets. Sample materials which are collected very often to test what it is the materials. Okay. So ports are inlets and outlets for collection of what it is the samples when the process is going on. What we call it the ports or the places. So the bioreactor, stirred tank bioreactor having agitating system, oxygen delivery system, foam breaking system, pH controlling system, and temperature controlling system, it is present. And there are the ports or the places, the samples are collected for testing of what it is materials also there. That's why it is a continuous culture system, that is agitator system, that is stirred tank bioreactor. So pH control for acid bases, steam for sterilization, this agitator system having a stirrer which are upper blades for breaking the foam, flat blade impeller for circulation of the culture broth, sterile air is passed through this. This is a hole, that is ring-like structure. Okay. In case of stirred tank bioreactor. So purpose of the upper blade breaking of what it is the foam. Acid base control, there is a one port, one place to maintain what it is acid base control. Okay. Sterile air, steam for sterilization. In case of first reactor, stirred tank bioreactor. So commonly used bioreactor, it is stirred tank bioreactor. So as it is cylindrical, curved at the base, helps in mixing up of the contents in the container. The stirrer facilitate even mixing oxygen availability throughout what it is container. and sparged stir tank bioreactor. The sparged stir tank bioreactor. And the word itself conveying sparging, that is forcing of sterile air. By forcing sterile air, what are created? That is, air bubbles are created. Once air bubbles are created, oxygen entrainment more. Where more oxygen availability, the fair chances of more products. These linked key points. The sparged stir tank bioreactor itself conveys its meaning that forcing of sterile air, air bubbles are created. As air bubbles are created, oxygen entrainment will be more. Once oxygen entrainment will be more, so there is a fair chance of quality the more product in case of sparged stir tank bioreactor. So in this, so oxygen air bubbles which are created, oxygen entrainment will be what it is more because of what it is sparging of what it is air bubbles are created, so oxygen entrainment will be what it is the more. The three key point: forcing of sterile air bubbles are created, oxygen entrainment will be more, so we can expect the fair chances of what it is the high productivity in case of what it is sparged stir tank bioreactor. Sparging is forcing of what it is sterile air. So in this two bioreactor, stir tank bioreactor, and second it is sparged stir tank bioreactor. Suppose there is a question in this. In bioreactor, biotechnologist forgot to add antibiotics. In bioreactor, biotechnologist forgot to add antibiotics. What change would you notice? Suppose it is the cell, it is recombinant DNA, the cells. So it is the host cell and it is RDNA. Now it is in bioreactor. If there is no antibiotic, so If there is an antibiotic, there is a tremendous pressure to what is recombinant. If there is no antibiotic, there is a great metabolic burden on the part of the host cell. The rDNA host cell tend to lose what it is rDNA if there is no what it is antibiotic. So we can compare this antibiotic as a boss in chamber. When boss present in chamber, all the substaff are going to be so attentive. If boss is out of the chamber, the substaff is not going to be attentive. They're going to be relaxed. It is just like this. So if the cells having what it is rDNA placed in a bioreactor, if he forgot to add antibiotic, host cell tend to lose what it is rDNA because it is a great metabolic burden to provide deoxynucleotides, DNA polymerases, energy. So great metabolic burden on the part of what it is the host cell. So host cell always tend to lose what it is rDNA. In order to maintain in case of the cells, in order to create a force, there must be what it is antibiotic. So antibiotic is comparable to what it is a boss in chamber. Because host cell tend to lose. Why it tend to lose? Because it has to provide deoxynucleotide, DNA, everything it has to provide. So the great burden on the part of the cell. So always tend to lose it. In order to prevent that, we are adding what it is antibiotics. This is the question. So in a bioreactor, biotechnologists forgot to add antibiotics. What change would you notice? The cells tend to lose recombinant DNA because there is no pressure, there is no antibiotic. Okay. So obtain this product and we have to extract this product. This product is not purified one. So we should undergo various processing before it is marketed. So the last step it is downstream processing. You got this product. The product it is extracted and it is purified. Before marketed. Before it is marketed, it should be extracted and it is purified. So up to this product, it is upstream process. After product has been obtained, after biotechnological product, biotechnological product, it was obtained. Extraction and purification, collectively what do you call it is? Downstream processing. So it is upstream processing and it is what it is, the downstream processing. So extraction and purification of biotechnological product before it is marketed, collectively what do you call it is? Downstream processing. So in this, by using preservatives, it is preserved, the product. That is what do you call formulations. By using preservatives. And clinical trials in case of drugs. So for obtaining any vaccine or the drug, it should thoroughly undergo the clinical trials, different batches of what it is the person, different aged person, that is young age, middle age and old age, to observe what it is its impact. So clinical trials in case of what it is the drugs. And quality control system to maintain its quality. So quality control system for every product is required, and downstream processing and quality control system varies from product to product, because it may be agricultural product, maybe it is veterinary product, otherwise it is food product, otherwise it is a medical product. So downstream processing varies from what it is product to what it is product. Downstream processing varies from product to product. So in all the cases, downstream processing is not similar. Okay, it is right, because it varies from product to product. So after obtaining the gene product, biotechnological product, extraction and purification collectively what they call downstream processing before it is being going to be what it is marketed. And you should undergo formulation by using what it is preservatives and thorough clinical trials in case of what it is the drugs, and strict quality control system should be practiced. And downstream processing varies from what it is product to what it is the product in case of downstream processing. So it is upstream, that is downstream. So downstream processing done after biotechnological product has been obtained. Okay. Insertional inactivation of antibiotics is one method of detecting recombinants. Question number 1. Question number 1. Answer. The question it is, insertional inactivation of antibiotics is one method of detecting recombinants. Question number 1. Yes, it is false. It should be what? Antibiotic resistant gene. Then it is going to be what it is, right? Okay, it is. Right answer, it is the antibiotic resistant gene. It is the right answer. Not it is antibiotic. Antibiotic resistant genes. In making rDNA molecule, following not required. In making rDNA molecule, following are not required. How many are not required? A option, DNA ligases. B, restriction enzyme. C, host cell. D, DNA polymerases. E, it is vector DNA. Question number 2. Question number 2. In making rDNA molecule, following not required. C and D or A and B, C or A and B. How many not required? Answer, message the answer. A, B. What is not required in making rDNA only? It is vector DNA. What is not required? Host cells which is not required for making rDNA. And DNA polymerase is not required. What is required? A, B, E required, not required. It is host cells and what it is DNA polymerase. Ligases which are required to link this DNA. Restriction enzymes are required to cleave the DNA, the source as well as what it is vector DNA. To express the DNA, to express the gene, what is required? Host cells which are required. That is going to provide what it is DNA polymerases. So for making rDNA which is not required, host cell DNA polymerase is not required. What is required? Vector DNA required. So in this required what? A, B, E. Not required what it is? C and D. Okay, it is. Required it is A, B, E. Because DNA ligase required to link what is the foreign DNA with vector DNA. Restriction enzymes which are required to cleave this source DNA as well as vector DNA. And vector DNA it is required to link what it is the foreign gene. So required it is A, B, E. Not required it is C and D. Okay, it is.